हेलो स्टूडेंट्स, आई एम मुस्कान। आज हम मेजर हिस्टोकम्पेटिबिलिटी कॉम्प्लेक्स MHC क्लास 1 एंड MHC क्लास 2 के बारे में पढ़ेंगे। हिस्ट्री। द पीटर गोरर इन 1930 found that for group of MHC molecule he used the blood sample of mice to identify the blood group antigen which is designated by first to fourth group of mice. The George Snell, Dorset and Berry received Nobel Prize in 1980 for their contribution to discovery of MHC molecule. Introduction. The measure histocompatibility complex is a group of highly conserved genes defined by its influence on tissue graft acceptance. The MHC refers to a cluster of genes responsible for immune response, transplantation antigen and protein of complement system. The MHC is a region of multiple loci which play major role in determination and transplanting tissue. Is accepted as self called histocompatibility and it is rejected as foreign called histoincompatibility. The antigen which is recognized by MHC is called transplantation antigen or histocompatibility antigen. The transplantation means the implantation of tissue from one individual to another. The implanted tissue is called graft. The MHC is refers to as the HLA complex in human and H2 complex in mice. The most study of MHC in mice and human. The three classes of MHC gene: class 1st MHC gene, class 2nd MHC gene and class 3rd MHC gene. The class 1st MHC gene, they encoded glycoprotein expressed on the surface of all nucleated cells, and the major function of class 1st gene product is presentation of peptide antigen to TC cells. The class 1st MHC molecule encoded by K and D region in mice and A, B and C loci in human. They first discover and they are expressed in the widest range of cell type. These are referred to as classical class 1st molecule. The additional genes or group of genes within the H2 and HLA complex also encoded class 1st molecule. These genes are designated non-classical class 1st gene. The class 2nd MHC gene, they encoded glycoprotein expressed on the antigen presenting cells, macrophages, dendritic cells and B cells. They present processed antigenic peptide to TH cell. The two chain of class 2nd MHC the two chain of class 2nd MHC molecule are encoded by IA and IE region in mice and DP, DQ and DR region in human. The class 3rd MHC gene, they encoded various secreted proteins that have immune functions including component of complement system and molecules involved in inflammation. The class 3rd product include the complement component C4, C2, BF and inflammatory cytokines including tumor necrosis factor TNF and heat shock protein. The class 1st and class 2nd MHC molecule are membrane bound glycoprotein that are closely related in both structure and function. The class 1st and class 2nd MHC molecule have been isolated and purified, and the three-dimensional structure of their extracellular domain has been determined by X-ray crystallography. The both type of membrane glycoprotein function as highly specialized antigen presenting molecule that form unusually stable complex with antigenic peptides display on the cell surface for recognition by the T cell. The structure of class 1st MHC molecule. The class 1st MHC molecule contain a 45 kilodalton alpha chain They associated non-covalently with 12 kilodalton beta 2 microglobulin molecule. The alpha chain is a transmembrane glycoprotein. They encoded by polymorphic genes within the A, B, and C region of human HLA complex and K and DL region of mouse H2 complex. The beta 2 microglobulin is a protein that encoded by highly conserved genes located on the different chromosome. The association of alpha chain with beta 2 microglobulin is required for expression of class 1st MHC molecule on cell membrane. The alpha chain is anchored in plasma membrane by its hydrophobic transmembrane segment and hydrophilic cytoplasmic tail. The enzyme papain cleave the alpha chain just 13 residue proximal to its transmembrane domain, the releasing the extracellular portion of the molecule. They consist of alpha 1, alpha 2, alpha 3 and beta 2 microglobulin. The purification and crystallization of an extracellular portion reveals two pairs of interacting domain. First is membrane distal pair. They are made up of alpha 1 and alpha 2 domain, and second is membrane proximal domain. They composed of alpha 3 domain and beta 2 microglobulin. The peptide binding cleft is located on the surface of class 1st MHC molecule, and it is large enough to bind a peptide of 8 to 10 amino acid. The alpha 3 domain appear to be highly conserved amongst class 1st MHC molecule, and they contain sequences that interact with CD8 membrane molecule. They present on TC cell. The antigen, which is recognized by TC cell, this is present in cytosolic pathway. The structure of class 2nd MHC molecule. They contain two different polypeptide chain. First is alpha chain 33 kilodalton and second is beta chain 28 kilodalton. They associated by non-covalently interaction. The class 2nd MHC molecule are membrane bound glycoprotein that contain external domains, a transmembrane segment and a cytoplasmic anchor segment. The each chain in class 2nd molecule contain two external domain, alpha 1 and alpha 2 domain in one chain and beta 1 and beta 2 domain in other chain. The membrane distal domain is made up of alpha 1 and beta 1, and membrane proximal domain is made up of alpha 2 and beta 2. The membrane distal domain portion of class 2nd molecule is composed of alpha 1 and beta 1 domain and form the antigen binding cleft for process antigen. The peptide binding cleft of HLA-DR1 is composed of floor of 8 antiparallel beta strand and side of antiparallel alpha helix. The class 2nd molecule lack the conserved residue that bind to terminal residue of short peptide and form instead on an open pocket, the class 2nd and open-ended groove. The class 2nd MHC molecule bind with peptide and present them to TH cell CD4+, an antigen which is recognized by TH cell, and this is present in endocytic pathway. The function of MHC molecule is to bind peptide the fragment derived from pathogens and display them on the cell surface for recognition by the TC cell. The TC cell in MHC class 1st molecule and the TH cell in MHC class 2nd molecule. The gene encoding the alpha chain of MHC class 1st molecule and alpha and beta chain of MHC class second molecule are linked within the complex, and the gene for beta 2 microglobulin and the invariant chain are on different chromosome number 15 and 5 in human and chromosome 2nd and 18 in mouse. Thank you.